I said there were 3 things that you had to know about that genetic code. And they were, you don't use universal. We know universal. That's the 3rd one. Unambiguous and redundant, exactly. And universal, yes, I know it's mostly, but it's pretty dang close. It's close enough that you can take the gene from an animal and put it into a plant and it works. You take the gene from a plant and put it into an animal and it works. In this case, how many people lived up north where there were fireflies, lightning bugs? All right, all right. Did you ever used to chase them around and catch them in jars and put them in your room as a nightlight? It's lots of fun.
Oh, that's so nice.
Yeah, what? Oh, I used to do that. I keep a jar of lightning bugs in the yeah.
See, that's more we can do that.
No, that's why you put holes in the jar and then you let them loose in the morning.
No, they always died in like 10 minutes.
No, mine survived.
My brother's
There, thank you. Grace's brother survived.
Yeah.
Yeah, but if you wanted to do something different to make light, you could take the gene that makes the chemical that makes their butt glow through the thin skin, and you could put that gene into a tobacco plant and make the plant make the protein so the plant glows. That's pretty dang cool. We got an animal gene in a plant, and the plant's like, oh, I'm supposed to make that. Okay, and it makes it.
How long would it take to make the plant glow?
Oh, this is a really good question. Now I'll give you the whole story, the true story, the honest story. The gene that they put into the tobacco plant is a gene for a enzyme called luciferase. Lucifer, the bringer of light, you know. Okay. So gene for luciferase, and the plant makes luciferase. But if you just saw it sitting out in a field, it would look like that. I mean, sorry, it wouldn't look like that. It would look like an ordinary tobacco plant. It would look. Tobacco plants are kind of like the white lab rat of plant.
But they don't glow it in the dark at night?
Right, they only glow if you give them the substrate that luciferase breaks down, because luciferase is an enzyme. So if you water them instead with pure water with a solution of the substrate in water, then they do that.
Okay.
But if you just see them out in the field, they just look like any other tobacco plant. But if at night you ran along with a sprinkler can or a big spray, then they'll glow. Yeah. I don't know. My guess would be no, because cigarettes are made out of dead tobacco plants. And if they're dead, those enzymes probably are all broken down and not working. Which is really sad, because you can sell them to all the raver kids really easy. All right. But anyway, so the same code is used, or a very, very similar code is used in all these things. Do we agree? A codon almost always has the same meaning if you put it into a different organism. It might, it could be a start codon. There's that difference. But that's why we can do awesome things like that. That's why we can make our fluorescent pets in the lab. Good stuff. Really good stuff. All right. So in the olden days, the way that we used to modify plants is that we would take a plant and we would selectively breed it to be different. For instance, wild potato plants, you're really not interested in baking those. They have potatoes about this big on their roots. And they're poisonous. Okay? But through selective breeding, we got rid of some of the alkaloid poisons, and we got the roots to be bigger and more starches, and now we have baking potatoes. All right. Same thing. We took one species of wild mustard plant, and they bred for, hey, I want it to look like, I want the flower buds. The flower buds are the tasty part, and you got broccoli. And then starting with the same plant, you breed selectively for, I like the leaves, because the leaves are awesome, and you get cabbage. And then you breed for, hey, that one there, I like the buds, except they taste horrible, so we'll make them taste like nothing. And that's how you get a cauliflower. And then they said, I like stalks, and that's how you get kale. And then they said, I want to make something extra disgusting, and so they bred for turnips. But this is all the same species. It's the exact same species of plant. They've just selectively bred it in all these different versions. Good? All right. So people have been manipulating genes in plants for thousands of years. Yeah, it's a test question. Thousands of years. It's just that we used to do it the old-fashioned way with selective breeding. Good? All right, so we're like, the way it worked is real simple. Hey, I like these disgusting turnipy roots. I want them to be bigger. So what do we do? Let's not kill that plant. Let's put it back in the dirt. We'll take cutting off of it and we'll raise it. Eat the others, but we're going to raise all offspring of this one, and the next season they all would have disgusting turnip roots, right? Right? So you go through a few generations and you have turnips. It's just selective breeding. You select which one gets to survive and breed, which one doesn't, right? All right, so artificial selection is the name for doing that. Artificial meaning it's not nature, it's people causing it to happen, right? So people in this old method of manipulating genes, they would breed animals and plants. They would say, I like these traits, so you get to live. I like these traits, so you go in the dinner pot. That's pretty simple. So you select for anything new or that you like, and over time you breed for those, and you end up with things that are similar to that. Good? Simple enough? All right. Before we go to that, let's just play the video. All right, so let's talk about Belgian blue cow. The first thing I want to point out, this was not genetic modification. They found cows that had a messed up allele, a messed up version of that gene, and they bred those. Because with that messed up version, there was nothing to say, hey, stop making muscle. So they made lots of muscle. Does that make sense? That is, I want you to understand it's not a guy in a white lab coat taking this gene and putting it into that. It's a guy in a white lab coat with a really bad job collecting sperm. Okay, it's a different job. What do you see here? This is a female Belgian blue. Yes, a C-section mark. There are issues with the breed having increased need to have cesarean sections when calving, which means increased cost, blah, blah, blah. These cows bulk up so fast with that messed up version of that gene that a lot of times they can't pass out through the mother's reproductive tract. They can't be born, many of them can't be born normally because they've gotten too big and muscly and they can't have the baby, and so they have to have a veterinarian take the baby out through her side. All right, the Belgian blue has very fast muscle growth. Would you agree? Yep. Some cattle grow really, really fast. They have this mutation that turns off the genes that regulate muscle growth, right? Right? Easy enough. And what do you do to make cows that look this way? You breed the ones with the mutation that turns off the muscle control. And then your muscles grow like crazy. Easy enough, right? It's still natural, artificial selection. Artificial because people are doing it. Selection because it's selecting who gets to breed and who doesn't. It's like, oh, you bulked up really fast, you get to breed. Oh, you didn't bulk up as fast. We're making hamburgers tonight. You would agree that you breed animals, okay, that have the traits you want. That's artificial selection. Now, people selected for a naturally occurring mutation, and they bred only the ones that had that mutation. The others they don't care about, right? And eventually they bulk up so fast you got to take the baby out with cesarean. All right, now we have the new way to manipulate genes. You don't look for, oh, this, and we'll breed these. You're like, I would like this gene to be there. Guys, this, you see the different colored stripes right here on the veins? This is a petunia flower with human genes in it. They're only expressing in the veins. Okay? Basically, we can get into this a lot more. There is a ridiculously obnoxious person. Yes, I'm inserting my personal bias here. Okay? I don't know him. He might be a really nice guy. But I don't consider him to be an artist. And he considers himself to be the world's first bio-artist. What he is a rich freak. Okay? And so he does, it would be different if he maybe made the petunia himself, put his own genes into it, but he didn't. He like, oh, my patrons have given me this money, and I will pay you to take my own genes and create a flower that has human genes in it. And they did. And that's the flower. Like, he also paid to have the first glow-in-the-dark bunny rabbit made and some other stuff like that. Yeah, we'll get to that. We'll get to that. All right. So Eduardo Cack is an idiot with a lot of money, but he pays people to put his genes into things, to put genes from X into Y. Now, I'm not against that. Obviously, I bought some fish in the lab. They're exactly that way. That have the yellowish, greenish ones. They have green fluorescent protein from jellyfish in them. Anyway, so I obviously am not against the moving of the genes. I just think it's stupid to call what he's doing art. It's not art. It's biotechnology. While we're at it, we'll just go with his other art projects first. There's Alba the rabbit. But what it was, it's the exact same gene that was put into it's a gene from a jellyfish. It's called GFP, green fluorescent protein. It's the same one that are in our little zebrafish in the lab. So, whatever. But it's taking a gene from this, putting in something that's only very, very distantly related in that it's an animal, and the gene still works, right? Jellyfish gene in a rabbit, it works. Jellyfish gene in a llama, it works. Jellyfish gene in whatever, and it works.
Do that hurt the animals when they put it in there?
No, it doesn't hurt them at all. They're not actually glowing. They call them glowing, but they're fluorescing, just like the chlorophyll that we work with in lab fluoresces. You excite it with this wavelength of light and it gives off another. That's what they're doing. Move on.
It's often the case that the least interesting stuff about science is the most discussed in general science media. Take, for example, the case of the glow-in-the-dark rat.
DNA technology, biotechnology, is a new way to control what genes are in organisms. We've been controlling what genes are in organisms for thousands of years by doing the selectively breeding. You are allowed to breed, and I'm so sorry for you. Now we literally say, hey, we like this gene, and we can put it into that organism or that organism, or so sorry, you didn't have it. You do now. We can do that. Actually, that wouldn't be true. They usually add it to the offspring that'll grow up to be that one, so you get the point. Anyway, so it's a new way to control organisms. We've been doing this since the 70s. We can turn a gene on that's normally off. We can turn a gene off that's normally on. Or we can add a completely new gene, like the jellyfish gene to the fish. Anyway, but now we can turn a gene on, turn a gene off, add new genes to an organism. Good? Good? I don't see a lot of nodding. Latavius, this making sense? All right, good.
I want like a 6.5.
All right, so here are examples for gene therapy. If a person has a bad gene, you can give him a copy of the good gene, and it could reduce or totally eliminate their symptoms. Now, we've had some problems with that, but we've had some big successes too. In fact, not the current issue, but the last month issue of Scientific American had a really good article called Gene Therapy's Second Act. Basically, well, we'll talk about all of this real quick. Gene therapy is a generic term. I should have put this in red. Know it, you have to know it for the test. It is genetic manipulation to reduce symptoms of a disease. I'll give you one where it worked really well. We talked before when we were talking about mutations about CF, cystic fibrosis. And remember, one of the things we said is that your lungs can get filled with really thick, nasty mucus you can't get out. Do you guys remember that? All right. Well, one of the things that can be done is you can take, and the reason people have that is that they have an allele, a version of the gene that doesn't make the right shaped protein. So the mucus gets really thick. If it had the right gene, you'd make normal mucus that lines the lungs, and that's real thin, and it's not a problem. So what do? You want to take this person who has the problem gene and give them a copy of the gene that's not a problem, right? So what they do, and this actually is working now. It's not like it's a widespread thing, but they've tried it and had success with it. They take the common cold virus. By the way, it's called the rhinovirus. You know, rhino nose, common cold, haha. Literally, the name of it's a rhinovirus. They take the virus and they take the nucleic acid core out of it. They take away the virus's genes that tell you to make new virus particles, and they replace it with the genes to make the right kind of mucus. And then the person with CF will literally, every so often, inhale intentionally a modified cold virus that it will do what it normally does. It will attack the lung cells. It will inject its core of nucleic acids in there and cause whatever it is to be made. Except that what it's causing to be made is not other viruses like normal. It's the right protein for the mucus. So it thins out the mucus. It reduces the symptoms. People are with CF are now surviving past their teenage years when it used to be that they never did. And one of the reasons why, admittedly we have better medical care now than we did 20, 30 years ago, but one of the other reasons is we can actually reduce the symptoms, which is a big deal. Now you just have them make at least some of the mucuses the right mucus, and that loosens up the overall and reduced symptoms. So there have been cases where it worked. There have been cases where it didn't. If I had read this article at the beginning of this semester, it would have been another assignment that you had to read it and write about because it's awesome.
Can we do it for extra credit?
No. There's no such thing as extra credit.
Yeah, I know.
All right. I just want to point out there have been some bad cases. And one of them was this guy, Jesse Gelsinger. They made modified viruses and they put them into his liver to treat a liver disorder. And the problem was that those modified viruses didn't attack the cells they wanted to in the liver. They instead, well, they did attack those, but they also attacked macrophages, which are white blood cells, they're part of the immune system, and dendritic cells, which are antigen-presenting cells, basically early warning sentinels for the immune system. And guess what? They didn't take kindly to being attacked by viruses, and they basically said, whatever's happening here, you need to stop it. And so he had this massive reaction by his immune system. Within 2 days, he was dead because they attacked his liver, because the liver cells now had the virus in it, and so, well, the modified virus, but you know what I mean. And so they attacked and killed his liver, and you can't live without one of those. So you understand that
I've had a couple.
that there have been some things where it worked and some things where it didn't. The whole point of the article is discussing why it didn't work well in the past, which we'll get into later on. I think I have some slides about. But the short version is that the viruses they were originally using for gene therapy, they inserted wherever they wanted. And if they inserted in the wrong place, do you remember we talked about spacer DNA, the junk DNA between genes? Yep. Okay, if you insert in there, so what? But if you insert in the middle of a gene that controls mitosis, poof, you got cancer. And there were a bunch of kids who they were treating who had a severe combined immunodeficiency disorder, which is like being born with HIV. I mean, it's not HIV, but it's being born with your immune system totally not working. And they figured out what genes were involved. Like, oh, we'll just give you the right copies of those genes. And they did, and it worked really well, and hooray, I'm surviving, and hooray, it's good, and I don't have to live in a bubble, it's perfect, and yay. And then all of a sudden, a couple of them came down with leukemia because they inserted in the spot where you control growth. And that's because they were using these little viruses called adenoviruses that they knew how to work with at the time. But adenoviruses insert wherever they happen to want to insert. And now we're using viruses that we can control a little better where they insert. Like they know this one attacks this chromosome. That's still not as precise as we'd like it, but it's a heck of a lot better. And this one attacks here, and you know, so modify those viruses, you have a little more control. Anyway, it's kind of interesting. Now, this is full of vocabulary words, this chapter. So you might as well write them all down. I tried to remember to put them all in red, but I obviously probably missed a few. But restriction enzymes. Let's talk about this. Would you agree that we don't like being infected by viruses? Okay. Oh, guess what? Bacteria don't either. And bacteria, even though they're one cell, they have their own little kind of immune system. They don't always know they've been infected by a virus, but if they do, they send out these enzymes called restriction enzymes, and they go along and they cut up the viral DNA, so it can't, or viral RNA, either one, because you know there are viruses that are DNA, there are RNA viruses, but it cuts up the nucleic acids of the virus. Hooray, works good. Well, scientists saw that and they were like, ooh, we could use that. So we saw these enzymes, these restriction enzymes, that's what we call them. And they're like, we could totally use those as scissors to cut up DNA. Like, say I want to take and insert a gene for green fluorescent protein. Well, you just pick a spot, and you're like, oh, let's cut there, and you cut it. And as long as you cut what you're inserting with the same one, it has the same ends. We'll get to that in a second in the slide. And you can then insert it, and they match up. And then you use DNA ligase, which we talked about before, being like the glue that puts the Okazaki fragments together. It will glue them together. And it worked fine. Which glue means hook the sugar phosphate backbone together. Are you with me, Mary? All right, good. All right, so restriction enzymes are how bacteria fight viruses. They cut the DNA into small pieces. They cut in specific sequences. If you had asked me last semester, I would have said the only ones that really matter to us, the ones we care about, are the ones that cut in a jagged manner and leave sticky ends. But what I'm about to tell you about is the newest and the greatest. They've only been doing this for 2 years, and it's even more awesome. But we'll get to that. Also, I might as well get this out of the way right now. One of the things that I emphasize on the test that I really want everybody to know is that we didn't invent most of these tools. We found them. We're going to talk about restriction enzymes. We found those, right? I'll talk later on about a high temperature polymerase. We found that in hot spring, a high temperature DNA polymerase. We found that in bacteria that live in hot springs. We're like, oh, we could use that. That'll work at high temperature. That'll be good. And so all of these things we found. But about 2 years ago, and I just found out about this semester, they started building their own synthetic restriction enzymes. Now that we know how restriction enzymes work, we can build better ones than nature did. And they call them zinc-finger polynucleases. And I'll talk about them in a second. So here, some restriction enzymes cut through both strands, right? And then some of them cut through one strand, break the hydrogen bonds between the strands, cut through the other strand, and leave an end that sticks out this way and an end that sticks out that way. You see that end that sticks out this way, end that sticks out this way. So for the longest time, we would say, oh, well, the restriction enzymes that leave the sticky ends, they're the awesome ones we use in biotech. And they were. And these, not so interesting, because the way it worked is these all want hydrogen bonds, right? And these all want hydrogen bonds. It's like Velcro for DNA. You could put, as long as you cut with the same restriction enzyme and have the same sticky ends, because you know the G has to go to a C, right? And the C has to go to a G. As long as you cut with the same restriction enzyme, you could take DNA from this species and DNA from this species, you could put them together and they would hold on to each other temporarily with hydrogen bonds, kind of like Velcro. Then you would put your DNA ligase in and it would hook the sugar phosphate backbone together, and then it's like one DNA molecule, like it was designed to be that way. That's how we would hook it together. Does that make sense? All right. What we have done now, by the way, the place of restriction enzyme cuts is called a restriction site. What?
That's time.
All right, hold on. Let me finish just this. And remind me, we are starting at restriction enzymes and zinc-finger polynucleases next time. No, I'm going to. Here, look. What? Okay, but this thing right here, this is the restriction site where the restriction enzyme cuts. What we're doing with these new synthetic ones that we're making are ones that look not for a little site like this, but the enzyme won't cut unless you match a 64, 128 base pair sequence. You see what I mean? It's like, because it's real likely that you'll find someone, like if you walk across campus, you say, I'm looking for someone whose middle name is Ann. You'll probably find someone, right? Right? But if you go across campus and you say, I'm looking for someone whose first name is Susan, her last name is Smith, and her middle name is Ann, it's less likely you're going to find that person. There are fewer. So when we're looking for this and this and this and this, and you make a long restriction site, that enzyme is very specifically targeted to attack and cut only one thing.
So it's
More specific.
more failed fruit.
Right. So the way that I'm going to describe this, restriction enzymes are like scissors for DNA. The zinc-finger polynuclease, which is a type of the synthetic ones that we're making, are like smart scissors that only cut in very specific spots that we choose to let them, instead of randomly cutting. Does that make sense? Yeah. That's okay. I've been bitten. Ow, he has teeth. Ow, ow, ow.